Dear listeners and viewers, I am Dr.K.Ramalingam. Uh, I did my research on basic science, basic medical science. I got my D.Sc degree for the investigations of the research done on animal models relating to human diseases. And I have been involved in cancer research for 10 years. In the last 10 years of my tenure of my service in Government Arts College, and also I did research in medical and biotech as a senior scientist. This lecture is in continuation of my previous talk titled I am not in conflict, war on cancer or war against cancer fails to win. This is the second part of the previous lecture, and it is also the concluding part of the previous lecture entitled I am not in conflict. I want to emphasize by these or by my lectures one thing or one secret, that is prevention is better than cure. Already I have appraised that the armamentarium in the therapeutic side against cancer and the treatment of cancer are surgery, radiation, chemotherapy mainly, and also the adjuvant endocrine therapies and monoclonal antibody therapy or the immunotherapy, etc. These therapies no doubt have brought a sea change in the treatment of cancer disease, and the chemotherapy has also brought a marked reduction of the tumor size and also the considerable or significant reduction in the number of cancer cells. I already said cytotoxic drugs in chemotherapy could kill 80 to 85% of the total cancer or tumor mass. But in spite of the above therapeutic procedures and clinical treatments, the cancers of the latter stages, so the cancer has got 4 stages, first stage, second and third and fourth stage. The latter stages means the third and fourth stage, which is very characteristic of a phenomenon called metastasis. So to cure or to contain this metastatic cancers, still 100% success has not been obtained by the clinical side. We have also seen in the previous lecture that cancer cells armamentaria against the therapeutic guns are also very formidable and strong, enough to resist the therapies. Cancer cells operate 3 methods of resistance, or they have got 3 modus operandi. Already I said that the first one is a genomic level operation of the cancer cells, the second one is the proteomic level operation of the cancer cells, and the third one is the metabolomics or metabolic level of operation of the cancer cells. Hannigan and Weinberg recently in 2011 have delineated 6 cardinal characteristics of the cancer cells. In his publication, he has delineated 6 cardinal features which are very common and observable in any type of cancer. The above fundamental postulates of Wiener and that is Hannigan and Wiener, when elaborately studied, the various cellular level and molecular level intricacies operating inside the cancer cells will become very clear or obvious. Here I am appraising you all some of the cancer mechanics that enable them to survive against the therapeutics or against the conventional therapeutic methods, namely the surgery, radiation and chemotherapy. This I do only to remind that the medical therapies should be revamped. This I emphasize because the medical therapies should be revamped with some alternative therapies. As a result, the success could be achieved to 100% even for metastatic cancers. The medical arena as well as the western side scientists have not agreed with the non-conventional herbal treatments, and they say that they have no clear-cut proof. I want to tell the listeners that the various cancer pathological sequences and the consequences become visible only after the manifestation. So the various cancer pathological symptoms appear in the body of a patient only after the manifestation of the disease, which takes a very long latent period of 15 to 20 years. That's why the suggestion that prevention is cure is very much appropriate for everybody to prevent the cancer. That's why doctors also will insist upon the prevention only, and the National Cancer Institute in USA also has come to agree that the prevention is the important point or important category when the cancer disease is concerned, when taken into consideration alongside the treatment aspects. Here I want to quote two Thirukurals of Valluvar, the Deiva Pulavar of Tamil Nadu. One Kural explains what will happen when one does not take the prevention, and another Kural explains what happens when one takes all preventive strategies or all preventions. The first one, that is, if one does not take preventions, for that he has written, Varumunnar Kavadan Valkai Yerimunnar Vaituru Vola Kedum. So Varumunnar Kavadan Valkai Yerimunnar Vaituru Vola Kedum. So here Valluvar explains that those are one who does not follow the prevention aspects, his life will be like a dust in the fire. The second one who takes precautions and preventions, for him or for them, Valluvar has written another second Quran, that is, Ediradar Kakum Arivinarku Illai Adira Varuvador Noi. Edirada Kakum Arivinarku Illai Adira Varuvador Noi. That is, when a man expects the future and when your man foresee the future, and such people will not, and if he takes precaution, he or they will not get any diseases. So these two are very impressive as far as the disease, general disease of the humankind is concerned, and also it is very appropriate for the cancer disease. Now I will read out the cancer cell adaptations and strategies for the survival mechanism. So cancer cells survive in spite of the formidable operations of three therapeutic procedures, radiation, surgery and chemotherapy. But in spite of that, the cancer cells are destroyed by 85%. But the remaining few residual cells of cancers are recuperating themselves, and they grow again in the primary site itself, and sometimes they become more virulent also, that they become more resistant also to the radiation, chemotherapy as well as for other chemical agents. Number one, cancer cells once after the transformation will express the tumor specific surface antigens. They are called TSSR or receptors, tumor surface specific receptors. Number two, these cancer specific or cancer cell specific surface receptors will bind to growth promoting hormones as well as for the growth promoting proteins synthesized by the cancer cell itself or from the outside, that is synthesized from outside. Number three, cancer cells exhibit also the cytoplasmic protein factors in the form of protein kinases, enzymes, which are responsible for the growth and proliferation of the cells. Number four, in cancer cells, the mitochondria are altered. The number of mitochondria as well as the profile of mitochondrial structures are modified. We have taken electron microscopic pictures of the mitochondrial structures, and they have got a varied type of profiles, and these mitochondria have got a mechanism to operate the anti-apoptosis phenomenon. That is, apoptosis means self-death or suicide of the cells, but cancer cells operate the anti-apoptotic mechanisms. This is done by the mitochondrial membranes. That is, the mitochondrial membrane regulates the exit of the calcium ions. If the mitochondrial membranes or the mitochondria inside contains the calcium ions, if these ions are let out through the mitochondrial membranes, these calcium ions will promote the apoptosis mechanism or the programmed cell death mechanism to the cell itself, cancer cell itself. That is why the mitochondria of the cancer cells operate the anti-apoptotic mechanism by preventing the exit of the calcium ions from the mitochondrial membranes. Number 5, the cancer cells through transcription and by translation synthesize several cell surviving proteins or the enzymes. So several enzymes and several proteins take part, and it is a very different profile, and these pathways operate one by one after another, or one over the other. As a result, the cancer cells were able to survive in spite of the therapeutic treatments in clinical site. Number 6, cancer cells synthesize the growth promoting prostaglandins. It is a hormone. It is a hormone called prostaglandin E. So the cancer cells synthesize the growth promoting prostaglandins E through an enzyme activity called cyclooxygenase number 2 activity, COX2. Shortly we call it as COX2 activity. There are two COX functions. Cyclooxygenase number 1 is favorable to the health of the cells, whereas the COX2 is detrimental to the cells. When COX2 operates, means it will promote the division of the cancer cells. Number 7, the cancer cells are able to protect or to regulate the glutathione concentration, and as a result of that signaling processes are going on inside the cancer cells. Number 8, outside the cancer cells they have got a matrix, and these matrix contain an array or a series of proteins. Some of the proteins are very important to keep the integrity of the cancer or proliferating or growing cancer mass. So these are the extracellular matrix proteins, and the same cancer cells could able to produce enzymes to clear away these matrix metalloproteins. They are called matrix metalloproteinase, and by clearing the matrix metalloproteinases, the cells, cancer cells from the primary site could able to move away from the primary site through the blood or through the lymph to distant organs. So this is the third stage. So the cancer cells start migrating from the primary site or from the epicenter to the distant organs. I have already said that the cancer cells during metastasis will go to different organs like liver, ovary, kidney, peritoneal cavity, brain, etc. etc. Number 10, the cancer cells exhibit metabolically both the anaerobic fermentation or glycolysis and also the aerobic glycolysis in order to get the energy supply. Then cancer cells have got, other than this anaerobic glycolytic mechanism, some other metabolic mechanisms like gluconeogenesis as well as pentose phosphate pathway is also operating inside the cancer cells. Number 12, production of lactic acid by fermentation and making the cancer cells interior and exterior into an acidic milieu is a characteristic feature. I already said that in cancer cells, they grow means they grow only in the acidic environment. So how this acidic environment is brought about means when it undergoes anaerobic glycolysis or fermentation, the lactic acid is formed. Then cancer cells employment of corecycle is another property. So the corecycle operation by the cancer cells and the resynthesis and recycling of the glucose from the lactic acid taken to the kidney and the liver is another mechanism to augment the cancer cells proliferation and growth. Number 14, extraction of other carbon sources. So if lactic acid is not available for the resupply of glucose for the cancer cell mass or the cancer dividing cell mass, then the cancer cells can able to pick up other carbon sources like glutamine, alpha ketoglutarate or the acetate compounds. So from these three, alpha ketoglutarate, acetate and glutamine, from these compounds they can take the carbon source for their energy production. Then important one is the HLA antigenic shift. So in our body, all cells have got a human leukocyte antigens, that is called MHC antigen, that is major histocompatibility antigens. And this particular protein or antigen is very important for transplantations. For transplantation operation, the compatibility of a donor and a recipient is verified only through this MHC complex or the HLA antigen action. So that way, when cancer cells started their proliferation and growth, they have a switching mechanism. They switch the HLA antigen from 1 to 2. So in all normal cells, non-cancerous cells, it is HLA1 is operating outside the cells. Whereas in cancer cells, that is why I said new antigens will be synthesized by the cancer cells. So the cancer cells will have HLA2 antigens. A new antigen is produced in order to escape from the human immunosurveillance mechanism. So that is another property of the cancer cell. So making devices to escape from the human immunosurveillance or the patient's immunosurveillance, another mechanism. So the 17th point is that cancer cells, I said 85% of the cancer cells are killed by the cytotoxic chemotherapeutic agents, but the remaining 15% are called residual cells, and they develop resistance mechanisms. So they develop the resistant genes. As a result, they could able to withstand the severe treatment or severe harsh treatment of the radiation as well as the chemical chemotherapy. And in addition to this, the tumor mass will have its own stem cells. Already in my lectures I told that every tissue or organ has got some stem cells, and these stem cells normally function to recuperate the lost cells. Suppose your cells are lost by means of any injury or by means of any infection, then these normal stem cells will replace the cell number, and as a result the organ function will be restored or reinstated. Likewise, in cancer cell mass or tumor cell mass also, 0.8 to 1% of the total mass of cells is comprising the stem cells, and these stem cells are different from the normal cancer cells in the expression of their surface antigens, in the expression of the proteins, etc., and also in their properties like invasion and metastasis. So the resistant cancer cells and the stem cells of the cancer mass are only responsible for the third stage invasion and metastasis to distant organs. Number 18, these stem cells and resistant cancer cells will not have any switching off mechanisms to undergo the apoptosis process, and they are not, already I said, they are not killed by the therapeutic mechanisms, and also they are not killed by the free radicals. They are not killed by the free radicals which most of the cytotoxic agents employ. So the resistant cancer cells are only responsible for the metastatic process. Even if chemotherapy cures to certain persons, among the patients, 30% of patients will have cancers will arise back once again, because these 30% of the patients have got some resistant genes, that is called the GYP gene. And if the patient has got that drug resistant gene, in the second instance of cancer, in his case, even the radiation and chemotherapy cannot help it. If they go for radiation and all, means again the normal cells will be affected and they become more vigorous cancer cells. Then in the metastasis of the resistant cancer cells as well as the stem cells, they make immunocompromisation. They make immunocompromisation with the distant organs. So there are, I have published one paper, how these metastatic cancer cells, when they reach an organ, distant organ, how they immunocompromise, and how it is just like a transplantation of an organ, a donor organ and a recipient organ, how it manages to fix it. So like that they have got some antigens. So that complementary antigens only will allow the cancer cells to survive in the distant organ. So there are two categories of antigens. One is a public antigen, another is a private antigen. So if the private antigen means definitely if we could find out the private antigen function, and if the private antigen could be employed as a treatment aspect, then definitely the secondary population of the cancer cells can be contained. Then the stem cells have got various anti-apoptotic pathways operated by the protein kinase enzymes. So the different types of anti-apoptotic pathways enable them to survive. In the last lecture, I insisted that both glucose and lactic acid formation in cancer cells should be prevented by the existing therapeutic modalities. But it is seldom possible until now. It is seldom possible or hardly possible until now. And my oft repeated saying is that restrict the supply of glucose. Not even the supply of glucose, the unstinted supply of glucose to the cancer cells through the food. So our food consumerism is only responsible for the cancerism also. I can say that the food consumerism is only responsible for the cancerism also. So we have to see how far we can control the glucose or the sugar supply in the cancer patients as well as to the cancer cells. So when we find out these methods, then we can say that we can reduce the severity of the disease in the patient, and we can also enhance the efficacy of the cytotoxic drugs and also the herbal drugs. The main item of the food rich in carbohydrates is the rice food. So conventional eating of the above rice in large amount has to be reduced to 1/10. Suppose if you reduce the intake of your rice by 1/10, think of it. So if you allow full amount, it will make the cells to 10 times proliferation. But if you reduce it, it will allow only 1 time proliferation. So that much difference we can bring it out. So in order to feel the stomach filling, the patient can go for other cereals rich in amygdalin or any other complex carbohydrates. The amygdalin rich foods like millet, jowar, ragi, traditional cereals like your samai arisi, kudarival arisi, like that kudarival rice or the sama rice, etc., can reduce the glucose level in the body. In the place of rice, the selected vegetables, I said that instead of taking more rice food, we can take more vegetables. And some of the cruciferous vegetables are anti-cancerous in action. Already I said the cruciferous family belonging vegetables like your cabbage or your broccoli or your cauliflower or your carrot. All these things are acting as anti-cancer foods. So it can be given to the patients. These can be given to the first patients. And the next one, the fish food which is rich in omega-3 fatty acids or essential fatty acids may be given in lunch or even in the breakfast alongside the porridge, that is kanji. So this I have already mentioned that in Japan the cancer incidence is very low, and in Hansa people the cancer is 0, and Nigeria is also coming under the zero cancer nation. So these people have got a habit of eating so much of anti-cancer foods like the amygdalin rich tapioca and the cereals other than the wheat or the rice. So this fish food, Japanese per capita consumption is 88 kilograms per annum. According to the latest statistics or data, Japanese the per capita consumption of fish goes to about 88 kilograms, whereas in Indian the per capita consumption is only about 19 kgs per year or annually. So this omega-3 fatty acids are very much important for not only for the sake of the requirement of essential fatty acids. Some of the essential fatty acids that is belonging to the omega-3 category can also revert back the cancer cells into normal cells. There are certain studies which have documented that certain essential fatty acids could reverse the cancer cells into normal cells. They can act upon the fatty profile of the cancer cell surface, and these fat rafts or the lipid rafts are transformed into normal cells lipid rafts. And the next one, it is before some 50-60 years, one doctor, that is Lady Dr. Budwick, she has treated 100 metastatic patients with her own diet, which includes the combination of the flax seeds plus cottage cheese plus curd. So those who are vegetarians, they can opt this very well, this Budwick's diet. So Budwick combined the flax seeds plus cottage cheese plus curd and treated 100 metastatic cancers, and she has also succeeded in curing certain patients in that group. So that is a very important diet every cancer patient should take into consideration, and every clinical specialist or oncologist should insist upon this Budwick's diet for their patients in hospitals. The above diet alters the cancer cell membranes, already I said, cancer cells membranes and fat rafts, and restore it to normal architecture. The actual mechanism involved may be necessitating further investigations. Then in between the breakfast and lunch, the patient may be given certain liquid foods, that is green tea or herbal tea or herbal extracts and herbal decoction, and this will restore the appetite. The appetite will be restored. Appetite will be enhanced by this green tea or the containing green tea and other drinks containing herbal extracts or herbal compounds. They also contain the necessary vitamins and minerals. And the glucose lowering effect is called hypoglycemic effect. It is found in both onions and garlics. So in the Liliaceae family in botany, the onions and garlics are coming. So both of them were found to have a lowering, blood glucose lowering effect. That is called hypoglycemic effect. So the two important chemical compounds present in the onions and garlic are, one is allyl propyl compound sulfite and another one is diallyl sulfite. So allyl propyl sulfite, and the second one is the diallyl sulfite. So they decrease the metabolism. So they decrease the blood glucose level by operating in the liver they operate at the liver level and they reduce the actions of the hexokinase and glucose 6 phosphatase activities. These two enzymes are only responsible for the replenishment of blood glucose and supply of glucose to cancer cells. Suppose if these onion compounds and the garlic compounds are given in the form of food or in the form of drugs, they will reduce the liver metabolism. As a result, the glucose supply will be restricted and the hypoglycemic effect over the cancer cells can be realized. Next one is the avoidance of salt. So when I say that glucose and sugars should be restricted, alongside sugars or glucose, the sodium salt also should be restricted, because in physiology we have already studied that the glucose and sodium are transported inside the cell at the same time. Simultaneously, the sodium ions and the glucose molecules will be transported inside the cells, normal cells as well as in the glucose cancer cells. So this mechanism is called symport. So by this symport mechanism also, the cancer cells can obtain the glucose alongside the sodium. So that way, if you reduce the sodium salt, you can reduce the glucose absorption by the cancer cells also. So that is another point which we have to take into consideration, and the medical side or oncological side also should insist upon the restriction of the normal salt. And next one, World War I and II only responsible for the emergence of your chemotherapeutic treatment modality, I said already. So the World War I and II have given two chemical agents to control the cancer cells. One chemical agent already I have discussed, that is called mustard agent colchicine, and the soldiers affected by this mustard gas were found to have a lower level of WBC's count, and the same principle has been applied to the leukemic patients, and they found that in the leukemic patients the abnormal WBCs have been reduced by this mustard agent, and when it is given in the form of a drug in 1946. Similarly, in the World War, another chemical employed was the chemical hydrogen sulfate. So it is given in the literature, medical literature, that another chemical in the World War employed is the hydrazine, HYDRAZINE, hydrazine sulfate, and this was used as a component for the rocket fuel during World War II. So during World War II, the hydrazine sulfate was used as a rocket fuel. And one researcher and Dr. Joseph Gold of the Syracuse Cancer Research Institute, New York, he found that hydrazine sulfate could inhibit the growth of the cancers like leukemia, lymphoma, melanoma, and other cancers like the liver cancers. And this hydrazine sulfate can even reduce the effects of leukoplakia and also other cancer types. So Joseph Gold proposed that hydrogen sulfate and various other hydroxides could contain the abnormal carbohydrate metabolism in cancer patients, both in the pre-terminal stage as well as in the terminal stages of cancer patients. And this hydrogen sulfate has got its hypoglycemic effect and cancer reducing effect in the following ways. It increases the appetite, number one. Number two, it decreases the pain. Number three, it diminishes the anorexia problem, side effect. Number four, it stabilized the tumor growth. Number five, it also promoted the survival of the patient. So according to the oncologist, the cachexia is the end result of after the metastasis. So cachexia is a process in which the patient is driven to skin and bones, and death is the end result of this cachexia. But according to Gold, he was of the opinion that these hydrogen salts could cut off a tumor's supply of new glucose or recycled glucose. Hydrogen sulfate could inhibit or could cut off the resupply of a new glucose or the resupply of the recycled glucose from the lactic acid from the liver and force the cancer cells to starve. So this point is of very important to take into consideration in the therapeutic site. So the phenomenon can put an end to this phenomenon of hydrogen sulfate action over the glucose will put an end to the cachexia. So according to the oncology books, the cachexia end process of the cancer continuum could not be replenished or could not be cured even by nutritional items, even by nutrition or nutraceuticals or nutritional chemicals cannot save the patient from cachexia. So that way they have written in the oncology books. But way back in 1900s, Mr. Joseph Gould, he has given this concept that hydrogen sulfate could be able to contain or stop or cut off the glucose supply to the growing cancer cells and their proliferation. And experiments were also done using this hydrogen sulfate. So they combined this hydrazine sulfate with the standard chemotherapeutic agents like cisplatin, vinblastine and bleomycin, and made the patients to take higher intake of calories. So when these conventional cytotoxic drugs are given along with hydrogen sulfate, it made the patients, the patients took higher calories of food materials. They showed greater survival. So this method of treatment of hydrogen sulfide, the combinatorial treatment of hydrogen sulfate with the conventional cytotoxic agents were tried for different types of cancers like liver cancer, lung cancer, stomach cancer, breast, Hodgkin's lymphoma, melanoma, leukemia, etc. So with these things, we can also say that what are all the ways by which the glucose supply could be restricted, we can try with that method also. In one reference I have seen that the anti-diabetic drug metformin was given to the animal models as well as to the patients. So when this hypoglycemic drug metformin was given to the patient, they observed the reduction in the tumor size. So this metformin drug is specific for diabetes disease, but when it was tried for the cancer patients, the experiments also, laboratory animal experiments as well as the human subjects also revealed that reduction of cancer or tumor mass happened with this metformin drug. So all these things again insist upon the importance of the glucose supply for the growth of the cancer cells, and if you stop the supply of glucose, the cancer growth also will be hampered or inhibited. So make the glucose non-available to both the patient and his cancer cells. So number one, make the glucose non-available to both the patient and his cancer cells. Make the glucose or sugar less available to the patient. Avoid the glucose-enriched pure carbohydrate food materials. Instead, take the complex carbohydrates. Take minimum carbohydrate food materials and take excess of vegetables endowed with or vegetables infused with certain important phytochemical compounds like the flavonoids or the beta-carotenes or the triterpenoids or the saponins. So in our research, we have found out in the herbal extract so many phytochemical compounds are present, right from monophenols, polyphenols, sterols, steroids, beta-carotenes, the flavonoids, flavones, then then other things are there. So many compounds, phytochemical compounds which are anti-cancerous in function. Already I said some of the phytochemical compounds like gingerin or your curcumin from the turmeric, they can arrest the COX-2 activity. The cyclooxygenase 2 activity is responsible for the cancer cell proliferation. When you take these herbal compounds, it will arrest the COX-2 activity. As a result, the proliferation will be contained. And all these herbal products work in unison. So you can see the same action with other herbal compounds also, like rutin. That is another compound which is a polyphenol, rutin. Likewise, the quercetin is again another polyphenol, quercetin. All these things will have the same action. That is, they will act on the enzyme which is enhancing the cancer growth. They will stop the enzyme functioning. As a result, the cancer growth could be contained. Then delay the glucose metabolism, delay the glucose absorption. So I said that in my book, Prudent Proxies, first volume, I said that for glucose, delaying the glucose absorption, the isabgol or the psyllium husk is a better choice. So by taking the psyllium powder, husk powder, or the isabgol powder, they will go and absorb the water. As a result, they will bulge. This isabgol or the psyllium powder, when they absorb, they will delay the process of glucose absorption in the intestine. So by that way, you can delay the absorption of glucose also in the patient. So the isabgol is now available in the form of a drug. So it is given for constipation. It is also given for some other ailments. But if the isabgol could delay the glucose absorption in the intestine, then through the blood it reaching the cancer cells also will be naturally delayed. Then another thing, burn the available glucose. The available glucose in the body should be burnt through aerobic exercise at least for 20 minutes per day. And take the fruits which bring alkalinity to the body interior. So I will talk about this alkaline milieu interior in the cancer cells, exterior and interior in the end. So take the fruits which are bringing alkalinity. For example, your amla is a very beautiful fruit. It is having dry amla and the wet amla. So the dry amla has got 600 times the more polyphenol compounds, and they will bring the alkalinity also. And those with the hyperglycemic index, already I said, take the metformin drug after the advice of your family physician. Then regarding the oil, I said already, the essential fatty acids are belonging to two categories, N6 category and N3 category. And N3 categories are favorable for the cure, whereas your N6 fatty acids are detrimental. Some of the N6 fatty acids like linoleic, already I said, linoleic is detrimental. And likewise, the palmitic acid is detrimental. So it will enhance the growth of the cancer cells. So choose the N3 fatty acids or the N3 essential fatty acids rich oils. So in that way, the wheat germ oil and the rice bran oil are very, very important. And in the rice bran oil, they have recently found one polysaccharide that is called RBS, that is rice bran saccharide. It was found to exhibit potent anti-cancer effect or anti-cancer activities. So it is very much advisable to use the rice bran oil for the patient. Then avoid oils with more N6 essential fatty acids. Instead, use oils with N3 EFAs. So I will list out what are the alkaline foods which bring the alkalinity to the cells, alkalinity to the cancer cells. So when your cancer cells environment is acidic, as long as it remains as acidic, it will go on growing or it will go on proliferation. So eat the alkaline foods. So I will read out what are the alkaline foods one can choose and given to the patients, or even to prevent the cancer. The normal people also can take these foods, namely the cucumber, then the beet green, soya tofu, green beans, onion, garlic, capsicum, pepper, sea vegetables, carrot, amla, etc. So these are the vegetables or the foods items we can select for making the cellular environment, normal cellular environment, as well as the converting the cancer cell environment into alkaline site. And the fruits side, the important fruits are watermelon, lemon, bananas, blueberry, apricot, pomegranate, berries, papaya, pineapple, avocado. So these are the alkaline fruits. They will induce the alkalinity inside the cancer cellular environment. As a result, the acidic milieu interior will be reduced. And take brown rice and other traditional varieties like sama, kudirivali, varagu, thinai, red rice, which are enriched with beta carotene. Beta carotene also very important phytochemical compound, which is responsible for the free radical fighting. So the free radicals injury only is responsible for the transformation of normal cells. So when your body has got more free radicals, the free radicals will affect the proteins, DNA, nucleus and cell transformation to cancer. So these are all the very characteristic features of the free radicals. In my book, Prudent Proxies to Prevent Cancer, I have given the various kinds of foods and vegetables or supplements which could prevent the buildup of sugar or glycemia in the blood. For example, the fresh vegetables belonging to the cruciferae family are good regulators of sugars. Then the compound flavones and flavonoids are good regulators of sugars. Monophenols from the tea, black tea, could complex with the glucose to form the polyphenols, which is also a good antioxidant, at the same time free radicals scavenger. So the polyphenols are good free radical fighters, and at the same time they can scavenge the free radicals, and at the same time they are the sugar regulators. And the glutamine rich foods, the glutamine is rich in the mushrooms. I have already given to the scholars that mushrooms, the butter mushroom and all types of, all the species of edible mushrooms have got their own characteristic phytochemical compound which will help to regulate the sugar content in the blood and the tissues. So these are all the food items which will reduce the sugar supply to the cancer cells, and as a result the patient's survival time can be prolonged to a certain extent with the help of the cytotoxic agents or the chemotherapeutic procedures. And with regard to cancer cell metabolism, I said that the lactic acid is the second promoter of the cancer cells proliferation. So lactic acid is formed in cancer cells both by the aerobic glycolysis as well as anaerobic glycolysis. Both of them yield only the same result. In the aerobic glycolysis also the end product is 2-ATP only. In the anaerobic fermentation also the end result is 2-ATP only. But this aerobic glycolysis used for the synthesis of the ingredients of cell division, namely the proteins, the DNA doubling, all these things are carried out. And there is a switching mechanism to anaerobic glycolytic site for the proliferation. So the already, as I said, the cancer cells recapitulate the embryonic character, at the same time they behave like the archaic organisms like bacteria, which are adaptable to the low oxygen environment. So the cancer cells also adapt themselves to live in a hypoxic situation. So this lactic acid production is another promoter for the growth and proliferation of the cancer cells, because by being acidic in nature, it will bring the acidic milieu interior as well as the exterior in the cancer tumor mass. So that is bringing an acidic milieu or acidic niche in the cancer cell mass is one character of the lactic acid. And another one, one study by Gatton B and Gillis in 2004, two authors, Gatton B and Gillis, in 2004, they have found out and observed that by acidifying the tumor microenvironment, the lactic acid has been shown to promote the invasion and metastasis. So the metastasis itself is promoted by the lactic acid, because lactic acid being a permeable factor, it can diffuse out of the cells. As a result, it will make the cells to enter into the blood as well as the lymph channels. That way the lactic acid promotes the metastasis or the migration or invasion of the cancer cells from the primary site. Number 3, the lactic acid or lactate also acts as a nutrient for some cells in the tumor. For example, the malignant cells in the less vascularized regions. So in the less vascularized regions, the malignant cancer cells of a tumor will use the lactate from the neighboring cells. From the neighboring cancer cells, they will draw the lactate. As a result, they will survive. So the lactic acid is also acting as a nutrient or a carbon source for the energy metabolism of certain malignant cells in the less vascularized regions. This is given by Sanvio et al. 2008. This is given by Sanvio et al. 2008. Then next one, lactic acid is responsible for the Cori cycle operation in the cancer patients. In the cancer patients, the Cori cycle operation operates only to promote the growth of the cancer cells, only to increase or enhance the proliferation of the cancer cells. That way, lactic acid is a promoter. And it is also a pro-inflammatory substance. Lactic acid is also a pro-inflammatory substance. And in the pathological conditions, we can see the lactic acid will enable the inflammatory changes. In our study, we have shown in a rat model study for arthritogenesis, the rats were injected with the complete Freund adjuvant, that is called a complete Freund adjuvant, CFA, as well as the incomplete Freund adjuvant. When this Freund's adjuvant is injected into the rats, it develops in the joints of the limbs the arthritic disease. The arthritogenesis occurs in the joints. And we have also observed the lactic acid levels. The lactic acid levels were up in these limb region muscles as well as in the tissue cells. And when we took the sections of the limbs in the joint region, we have found out that the T lymphocytes and macrophages are nearby. So in the joint regions, the T lymphocytes and macrophages are sitting, and they do the damage. So this is these two cells, the macrophages eat away the cells. So this is how come these macrophages and T lymphocytes can come to that joint region, means from the peripheral blood circulation. The lactic acid permeability factor may enable the entry or exit of these the pro-inflammatory cells, namely the macrophages and the T lymphocytes in the joint regions in the animal model, rat arthritogenesis animal model. So these are the important points with regard to lactic acid. And how to contain this lactic acid or acidic milieu interior, I have said by nutrition we can contain the acidic milieu interior. By nutrition means taking the proper vegetables, by taking the proper food items, and we can bring down by taking the proper medication, we can bring down the glucose supply to the cancer cell mass as well as the lactic acid formation. And at the same time, in recent years, the pandemic outbreak of COVID-19, everybody knows, everybody is now aware how come this, how this SARS-CoV-2 is damaging the whole world. The whole world nations are suffering due to the attack of this SARS-CoV-2. So the severe acute respiratory syndrome virus is bringing damage to the lung tissues, As a result, the person dies due to respiratory illness. That is why it is given the term severe acute respiratory syndrome. So the people are dying because of this severe acute respiratory syndrome. And in that period last year, some researchers have also elucidated the actual mechanism by which this SARS-CoV-2 enters into the lung alveolar epithelial cells as well as the lung endothelial cells. They have found that this entry of the SARS-CoV-2 viruses is enabled by one factor that is called angiotensin controlling enzyme 2. So this angiotensin controlling enzyme 2, that is called shortly ACE2, it is released from the kidney. So ACE2 is released from the kidney, and it goes to the lung epithelial cells and endothelial cells where they act as a receptor. And in this angiotensin controlling enzyme 2 acts as a receptor, and it allows the entry of the pathogen. It allows the entry of the SARS-CoV-2 virus inside the endothelial cells as well as inside the lung alveolar epithelial cells. Can you follow? So the angiotensin regulating enzyme 2 released from the kidney reaches the lung where it acts as a receptor over the surface of the epithelial cells as well as the endothelial cells and allow the entry or internalization. That is called internalization. So when the infective agent enters inside the cells, it is called internalization. So this internalization of the virus is brought about by this ACE2, and they have also revealed that initially in the last year period, researchers have established that certain drugs like the hydroxychloroquine or chloroquine and azithromycin and zinc containing drugs could able to prevent the infection, because these azithromycin and zinc and this hydroxychloroquine will act as the receptors and prevent the entry of this or internalization of this COVID-2, and this will enter these drugs as well as the zinc enters inside the alveolar epithelial cells, and they will neutralize the acidic environment. So inside the alveolar epithelial cells, if already the COVID-2 infection was there, these drugs will neutralize the acidity. As a result, they will bring the alkalinity. So they bring the alkalinity inside the alveolar epithelial cells, and as long as the acidic milieu interior is present inside the alveolar epithelial cells, the virus multiplies and the viral bodies multiply and their protein undergo glycation or glycosylation in the Golgi complex of the alveolar cells. In the Golgi complex of the alveolar cells, there are different types of sugars like glucose, fructose, galactose are present, and this viral protein will go and bind with this glucose or galactose or fructose, that is called by a process called glycosylation. As a result, only the assemblage of the bacteria or the assemblage of the viruses SARS-CoV-2 occurs and the damage comes, lung damage comes. But once these drugs enter inside, they will neutralize the acidity and they bring an alkalinity. As a result, they can stop the RNA multiplication of the RNA of the viruses, RNA multiplication, viral RNA multiplication, at the same time the protein formation inside the cells. So this research information gives a cue for the cancer cells also. So when the cancer cells are also thriving only under the acidic milieu interior, so when such trials were made in research in future, such trials are made in future where these drugs may also bring about the alkalinity inside the cancer cells, and they can also prevent the multiplication of the protein or the synthesis of protein. They will inhibit, they will prevent the DNA replication, RNA replication, etc. So this is a beautiful observation given by the researchers in Western countries, how this mechanism of infection of SARS-CoV-2 occurs and the COVID-19 disease is manifested and lung pathological changes occur in the patient and how he dies and all. So this is a very beautiful information which gives a cue for the cancer treatments also. So with this I try to conclude again. I read the last sentences, the last sentence of my previous lecture, that is lives and reputations depend on the full adherence to good practice. Lives and reputations depend on the full adherence to good practice, and this is also applicable to good medical practice. And the concept, before saying thank you, a decision is for a vision, a decision is for a vision, a vision is for a mission, a vision is for a mission, and a mission is for an endeavor of success. Cure cancer, win the cancer war. Thank you.